Natural selection is a major force driving allele frequency change. Natural selection. It is the major force driving allele frequency change. It is also the chief mechanism for transforming populations. It is the principal force that shifts allele frequencies within large populations. It is discovered independently by Charles Darwin and Alfred Russel Wallace. The Wallace-Darwin concept of natural selection. Individuals exhibit variations in phenotype. Variations are heritable in that they can be passed on. Organisms tend to reproduce in exponential fashion. More offspring are produced than can survive. But some phenotypes are more successful at their survival and reproduce at higher rates. Fitness is symbolized as W. Hardy-Wimber analysis allows fitness W to be examined for each phenotype. Fitness is an individual's genetic contribution to the future generations. Homozygous recessive individual who dies before producing offspring, it is 0 in fitness, which is W 0. Frequency of recessive allele will decrease in each generation. As you can see, if it is not fit, by generation A will be going down, and in the end A will disappear because it is unfit for survival. Degrees of selection. Outcome of different degrees of selection against non-lethal recessive allele A. Density of selection varies. Strong red curve to weak blue curve. Intermediate you have yellow, purple and green curves. Selection for traits can be classified as directional selection, stabilizing selection and disruptive selection. Directional selection means phenotypes at one end of the spectrum become selected for or against, usually as a result of changes in environment. For example, the beak size in finches during dry years increased due to strong selection. 1977, the mean beak depth in millimeters for dry year is 9.8, and then after the dry year, the mean beak depth has gone down. And then in 1980, which is also a dry year, the beak depth increases, and then it goes down again. 1982 and 1984, as you can see, there's a difference because in 1982 it is a dry year, so beak depth will be quite high. But in wet year, in 1983, it goes up again. And after that, it goes down again. Stabilizing selection. In stabilizing selection, it favors the intermediate types. The extreme phenotypes are selected against, meaning that they will be lower. It reduces population variance over time but not the mean. For example, the human birth weight study over an 11-year period. So as you can see here, the birth weight for pounds, the mean will be 6 pounds, or how many kg? How many kg is 12 kg, right? So the infant births will be highest at the mean, which is the 6 pound weight. If a baby is lighter, there will be more deaths. More deaths after 3 pounds, and then 2 even higher, and then 0, meaning that if the baby is so light, then they'll be mortal. Also, after the mean, the infant birth will be lowered, meaning that if the baby is too big, it will increase the mortality. The mortality will be increased when after 10 pounds, and then after that there'll be no more. There'll be no more babies that has been born over 10 pounds. Disruptive selection. Disruptive selection is the opposite of what we have just learned. So in disruptive selection, the intermediate is not good for survival. They are selected against. So you must be either in one extremes, either dark or light, short or tall. If you are in the middle, if you are medium, then you will be selected against. For example, the low and high bristle number in Drosophila selection. Okay, so usually for females, the females in Drosophila population would select any males which is either having a lot of bristles or short bristles. They will not look for the medium bristles. After many generations, you can find out that they are either the longest bristles or the shortest bristles. There's no in between. Mutation creates new alleles in the gene pool. Within a population, the gene pool is reshuffled each generation. Mutation is the only process that creates new alleles in gene pool. Because if you have a pack of cards, you shuffle and shuffle, you still have the same cards. What if one of the cards you draw or you put some watercolor on it, it becomes a new card? So mutation is like that. It creates new alleles in gene pool, and most mutations are recessive. Indirect methods using probability or statistics or large-scale screening problems are often employed to estimate the mutation rates. The mutation rates, it is the number of new mutant alleles for a given number of gametes. If the mutation rate is known, the extent of change to the allele frequency from one generation to the next can be estimated. Achondroplasia. It is a dominant form of dwarfism in humans. It is a skeletal disorder, meaning that it affects your bones. It causes your skull to be enlarged compared to the body, short arms and legs, diagnosed with X-ray examination at birth, and it is fulfilling the requirements for measuring mutation rates. Next section. Migration and gene flow can alter allele frequencies. Migration occurs when individuals move between populations. Species divides into populations that are separated geographically. Allele frequencies in new populations may differ over time. So here is the genetic history of Europe. Genetic drift causes random changes in allele frequency in small populations. Genetic drift. Genetic drift is a significant random fluctuation in allele frequencies in small populations. It is possible by chance alone. The degree of fluctuation increases as population size decreases. It can also occur as a result of founder effect or genetic bottleneck. What is a founder effect? So, founder effect means that if you are the first population, if you are the first individual in an area which has no other alleles, you are the founder. Meaning that your genes will be the original gene. Even though you come from a bigger population, in the bigger population you might have people who are different from you. There are many different alleles, many different variations. But because you are alone in that new area, you are the founder of all of your populations. For example, if the Earth has been destroyed by a nuclear war, the survivors might make a new population, might might make new cities, new villages, but the new population will not be the same as the previous population before the war, before everybody died. Okay? Genetic bottleneck. It can also cause genetic drift. It is when a large population undergoes drastic but temporary reduction in numbers. For example, if there is an accident, if there is a forest fire, if there is a meteor suddenly striking the area, if there is a disaster, okay, disaster would wipe out almost all of the population, right? Populations may recover, but with greatly reduced genetic diversity. Example found the effect in human populations. So in the Navajo are a tribe in the deepest part of America. OCA is actually a disease where you have lack of melanin pigment in your eyes. So you have oculocutaneous. Oculo means eye, cutaneous means skin. So you have albinism in your skin. Okay, but not just that, you also have albinism in your hair and iris. So mean that your eyes will be a little bit lighter color, your hair will also be white, your skin will also be white, mean that you would be very susceptible to cancer if you are exposed to sunlight as an OCA person. The PCR screening of DNA and analysis of OCA frequency indicate that the mutation allele might have risen as a founder mutation 400 to 1000 years ago. Here's a picture. So here you can see the big A, small a, and big A, big A, right? Now both of these persons would have the disease, this AL big with this small a allele. Small a allele can be inherited. So the chance that this female will inherit 2 copies of her great-grandmother's allele, A allele, is F 1/2 minus how many generations? Okay, minus 1/2 times 8 generations, you get 64. Because the female's 2 allele could be identical by descent from any of the 4 different alleles, so F is 4 times 1/64 is 1/16. So as you can see here, if this woman marries somebody from her own lineage, there will be a big chance for her to inherit the allele from her great-grandmother. Nonrandom mating changes genotype frequency but not allele frequency. Nonrandom mating, it can change frequency of genotype in population. Sequence selection for or against certain genotype can affect overall allele frequencies, but non-random mating itself does not directly change allele frequencies. Forms of non-random mating: positive, negative, and inbreeding. Positive assorted mating: similar genotype more likely to mate than dissimilar ones. Negative assorted mating, just like the name says, dissimilar genotypes are more likely to mate than similar one. And inbreeding, the mating individuals are related. Inbreeding is the mating among closely related individuals. For a given allele, inbreeding increases the proportion of homozygotes in population. And completely inbred population theoretically will consist only of homozygotes. F, or coefficients of inbreeding. Quantify the probability that 2 alleles of a given individual are identical because they are descended from a single copy of allele in ancestor. F equals 1. All individuals in population are homozygous. Both alleles come from same ancestral copy. F 0. No individual has 2 alleles derived from the common ancestral copy, meaning that they are not inbreds. Reduced gene flow, selection, and genetic drift can lead to speciation. Species and speciation. Species is a group of actually or potentially interbreeding organisms that is reproductively isolated in nature from all other such groups. Speciation, associated with changes in genetic structure or population with genetic divergence. So speciation means the creation of species from one that used to be part of a bigger population. Now it becomes its own selective and separate population. The divergence of population can reflect action of natural selection, genetic drift, or both. Macroevolution is genetic changes that result in reproductive acceleration between or among populations. The evolution becomes so large that the original and the new population cannot breed with each other anymore. So it leads to formation of the new species. So there is something that's called reproductive isolation, meaning that both the populations can no longer reproduce with each other. They are isolated. The reproductive isolating mechanisms are biological barriers that prevent or reduce interbreeding between populations. Mechanisms may be ecological, behavioral, seasonal, mechanical, or physiological. Isolating mechanisms mean that whatever that can separate them both. Prezygotic and postzygotic. Prezygotic, it happens before the fertilization. It prevents mating from taking place after all. But what if they can mate but still they are unable to breed? So we call it the postzygotic isolating mechanisms. So even though the two individuals mate with each other, there are still some things that prevent babies from being born or prevent a new population, a hybrid, from prospering. Changes that lead into speciation. The first one is the geographic changes. For example, the ancestors of the snapping shrimp. Prior to formation of Isthmus of Panama, members were of single species, and so the area where they live used to be an open sea. But after the sea level receded and the tectonic plate moved, now there is a border or boundary in between them. So in between the seas, there's something that is dividing the sea into half. It is the Isthmus of Panama. It is a long stretch of land and that separates between the two seas. So just like us in Malaysia, we have the north. The north we have Patani. We have the Isthmus of Kra, which is the border between Thailand and Malaysia. Okay. That area has a very narrow strip of land. So it is the land that separates the two seas. When the isthmus closed, each of the 7 ancestral species were divided into 2 separate populations. So this is the shrimp. Section 10.9. Rate of macroevolution and speciation. So average time for speciation means that how long does it take for a new species to take over? 100,000 to 10 million years. So it takes a long time for a new species to emerge. But there are some cases where species actually became new species very fast. We call it rapid speciation. Such as fishes in the East African lakes, such as the marine salmon, palm trees on Azores Island, polypod plants, and also brown algae in the Baltic Sea. So, for example, the lake in Nicaragua called the Lake Apoyo. It is formed within the past 23,000 years. It is the host of 2 species of cichlid fish. So cichlids, they are famous for keeping their young fishes, their children within the mouths. Okay, so mother cichlid is common in the region, but arrow cichlid, it is found only in this lake. So Lake Apoyo is a very isolated lake that is not part of the bigger mainstream lakes. Because Lake Nicaragua is the main lake, and then you have the Lake Malagua, which is the bigger one. But Lake Apoyo is kind of like small and kind of recent and kind of separated from the bigger ones. So arrow cichlid, they look kind of different compared to the minders. So scientists has checked it phylogenetically, morphologically, ecologically, and sequence analysis of mtDNA, meaning the DNA from the mitochondrion, indicates that both of these are from the monophyletic group, meaning that both of them are related. So it is concluded that arrow cichlid evolved from the Midas cichlid. So the Midas cichlid was the original, and from there it evolved into the arrow cichlid. So A and B. So you might recognize the picture in A. The picture in A is actually called in Malaysia and also China as flower horn. The flower horn, it is a quite expensive fish. It is ornamental. The bigger the bulge, it means that it can bring you better luck. So that's what people will think. Yeah, so that's what's called the arrow cichlid. But for the Midas cichlid, which is the original, look at its color and look at its forehead. It doesn't have a big forehead. The colors are kind of drab, and the body is much more hydrodynamic. So they can really swim fast, and they do not attract predators. But for the arrow changeling, look at how it look like. Very colorful, very bright, attractive. So for any predators, they're easy to spot. Next section. Phylogeny can be used to analyze evolutionary history. We use genetic differences among present-day species to reconstruct evolutionary histories of phylogenies. For example, there's a tree, figure 25-21. The branches in phylogenetic trees represent lineages over time. Monophyletic group consists of ancestral species and all its descendants, meaning that all of the organisms that is attached to this branch of a tree, we can call them monophyletic. So you have the root and the branch. Okay, and then you have the node and the tip. In the tip, you have species A, B, C. Species B and C are connected by a node. The species B and C, the node actually shares the same branch as species A, so meaning that B and C are monophyletic. But back in time, further in time, species A, B, C are actually coming from the same type of ancestor. Cytochrome C. Cytochrome C is a eukaryotic mitochondrial protein. Phylogenetic reconstruction of amino acids of cytochrome C indicated that it has evolved very slowly in a variety of organisms. So what is genetic equidistance? It is a difference in amino acid sequence between species which are proportional to evolutionary distance. So if you look at the sequence, the closer the sequence means that it doesn't actually evolve a lot. It doesn't travel far. The bigger the distance, it means the species has evolved very much. Minimal mutational distances in amino acid differences in protein. So data on distances between cytochrome C genes on 19 organisms were used to construct a phylogenetic tree showing relationship among various species. So here is the phylogenetic tree. If you're going to compare yeast, the simplest one, with us, human, there is a lot of distance. 515.2, 5.4, 4.2, 3.3, 1. There's a lot of distance from our gene to their gene. But if you look at our gene with monkeys, there's not a lot of distances, 0.8 to 0.2. So we only differ from 0.6 in terms of distances. If you look at the bird species, pigeon, ducks, chickens, penguins, all of them are a little bit kind of closer to each other. If you want to compare with fishes, the tuna, and then up we have the snake. But turtles are even more evolved compared to snakes. And then what else? You can see the dogs, much more evolved compared to horses and donkeys. And then pigs, then rabbits, kangaroos. So compared to all of these, dogs are the most evolved in terms of cytochrome C. Molecular clocks. Molecular clocks measure rate of evolutionary change. It measures in terms of amino acids or nucleotide sequences, evolutionary changes accumulate at constant rate over time. So in figure 25-22, you can see the molecular clock showing divergence time for humans and other vertebrates based on fossil record. So as you can see, divergence time based on fossil records, for synonymous nucleotide substitutions is very tall gradient compared to amino acid substitutions and non-synonymous. If you want to compare all three of them, synonymous nucleotide substitution is the most mutation that happens for year by year compared to amino acid substitution and nucleotide substitutions. So to cover origins of our genome, genetic diversity in empty DNA is used to infer where Homo sapiens originated. So there is a high level of diversity in African populations, but non-African populations had origins about 50,000 years ago. So the Homo sapiens originated in Africa from early species of Homo. Neanderthals, our closest relatives. Neanderthals lived in Europe and Western Asia about 300 years ago, but they disappeared about 30,000 years ago. So it coexisted with anatomically modern humans, Homo sapiens, about 30,000 years in several regions. So the phylogenetic tree shows pattern and times of divergence of Neanderthals and our species. Neanderthals are not direct ancestors to our species. Neanderthals and members of our species interbred for time, and they contributed to our genome. Okay, so in terms of the genomic data, humans and Neanderthal have split into two. the most Neanderthal remains would end at 28,000 years ago. But before that, the earliest modern humans in Europe was found 41,000 years ago. So here is the phylogenetic tree. So from the ancestor, you have the potential unknown hominin, not known yet. Then from there, you have the Denisovan and Neanderthal. Neanderthal first, and then Denisovans. And then after that, later would be us, the modern humans. For the modern humans, you have the Oceanians, the Asians, the Europe, and the Africans. Okay, so in between them, Africans are the most purebred of all humans, because the African population of Homo sapiens do not intermix with other type of humans. But in terms of modern humans, the modern humans actually mix with Neanderthals, so we have some of these genes. So from there, we have 3 lineages of modern humans: the Melanesians, the Han, and the French. So the Melanesians actually have mixed with the Denisovans from Siberia during their migration. So mean that the Melanesians actually have some Denisovan genes together with Neanderthal genes within them. So this is because of interbreeding. So the Han and the French only has the Neanderthal genes but no Denisovans. The end.